My name's Sean McGuire. I'm moderating this session. I work for the Public Water Supply section in DEQ. So if you drink municipal water, we're the ones that regulate you, your water supply. For those that are thinking about session hopping, don't know. If you would, we're going to follow the presentation list as it is in the app, not on the board. So our first presenter is Dr. David Johnston, who is an assistant professor of the practice of marine conservation and ecology at Duke University. He holds a PhD from Duke and received postdoctoral training at the Monterey Bay Aquarium Research Institute in California. His professional experience ranges from leading research programs for NOAA to working as an ecologist with the NGO sector. So he's our first presenter. Thank you.
Thank you for the introduction, and thanks everyone for taking your time out to come and join the session and listen to me give you some background information on some of the research we're doing. I'm also the director of our new UAS facility at the Duke Marine Lab, and so we've been running for about 2 years now. And I want to give you just sort of an overview of some of the stuff that we're working on and how that links into spatial analysis of marine and coastal systems. So on deck, the first thing I want to talk about is kind of make a pitch to you about why drones are actually really crucial or useful for us in the marine context, and then give you guys an overview really quickly of our current capabilities in terms of aircraft and sensors, what we're working on. And then I want to present sort of just 3 really quick case studies on how we're using drones to do population assessments, how we're using structure from motion techniques to reconstruct beaches, barrier islands, and then very quickly illustrate a new project that we just got funded, mapping out some wrecks in a really interesting place on the Potomac River. So a very broad range of things and indicative of the kinds of things that we're working on. So here's my pitch to you guys. For me, you know, as a marine scientist, if you look at this cartoon at the bottom, you can imagine that that's the little Duke Marine Lab there at the bottom, and across the creek is our Rachel Carson Estuarine Reserve. And traditionally, we would go across there with quadrats or with maybe a laser line scan system, and we would make very, very detailed measurements. We would learn a lot about the ecology from seagrass all the way up to seabirds. But then really the only other thing that we would have, after that was satellites and aircraft, occupied aircraft. And as you guys probably all know, there are challenges in using both of those in terms of logistics, costs, and things like atmospheric interference. And along come drones, which essentially put us right in the middle of that and provide us very, very high resolution and most importantly, on demand. So we can go and fly in almost any condition, up to 22 knots of wind and collect really good data. We can fly in the rain. And that allows us to do sampling before and after events and a move away from just change detection to understanding process. So very useful. A couple of other reasons why it's important for us as marine scientists. Paper published in 2003 looked at the mortality factors for wildlife biologists, and 66% of those were related to aviation incidents. So I'm not saying we need to get rid of occupied aircraft, but there are ways that we can reduce human risk by taking advantage of drones. We also can do it cheaper. In some cases, for example, we just conducted a series of population assessments of seals for the Canadian Department of Fisheries and Oceans, and our costs came in at 20% of what they had projected for the occupied aircraft that they would need to be able to do that survey. So in this case, a good example of how we might be able to reduce costs and risks. So that's my pitch to you guys about why I feel like this is so important for us to be able to develop. So I said we've got a UAS facility now. We actually just got an NSF grant to renovate that facility, boost our computing infrastructure, and actually purchase new aircraft, platforms, and sensors. And so right now we've got 5 fixed-wing aircraft that allow us to map at 2.5 centimeters per pixel on the ground, RTK corrected, a suite of multispectral sensors and RGB sensors and thermal sensors, aircraft that are autonomous. We can land in the water if we need to. And reaching up to 2 hours flight time, so for some of our aircraft. On the multi-rotor aircraft, we're achieving sub-centimeter resolution on surveys. Again, a suite of different sensors from multispectral through thermal, streaming video, really great for doing animal behavior, things like that. I'm not going to talk about those today. And achieving up to 40-minute flight times with some of our heavy lift multicopters. So getting some really great persistence in the air. The first example that I want to talk about is us working on gray seals in Canada. And I have a short video that will give you an idea of what the habitat's like that we're working in and the software that we use to be able to fly and one of our fixed wings and do our mapping missions. And so this little quick video is going to show the habitat. It's going to illustrate setting up a very quick mission. It's going to show launch and recovery of the aircraft. That'll give you a flavor for how useful it is. In this case, you can get on site, set up your flight plan, and execute your survey in a really, really short period of time, 10 minutes maybe to set up a survey, and that allows us to hit weather windows that we would not normally be able to access with other methods. So here we go. It's a little bit in the helicopter. In this case, because we're restricted to line of sight, we weren't able to fly to the island and map it, but we're working on that. Get to the habitat. It's working with gray seals in the springtime, so it's still pretty cold. I'm from Canada, so it's pretty normal to have snow in the spring, but not down here. We set up our approach vector for landing, our region of interest. We upload that to the drone, and it's ready to go. In this case, it's a SenseFly EV. We set our ground resolution in the software. We shake it 3 times, pitch it in the air, off it goes. Monitor it in flight. This gives you an idea of the footprint on the ground for the photos, so we're able to assess coverage as we're conducting the surveys. And a quick set of examples of photos, individual photos that are taken. These black blobs are seals that you see. There are also seal pups that are white, and I'll talk a little bit more about that in a second. The aircraft comes back, senses wind direction, and chooses an approach vector within that pie that we set to land most efficiently into the wind. Here we come in. It's, I guess, the Air Force would call that a hard landing. We often catch them now. But a very, very simple set of parameters. Like I said, very quick to get on site and be able to do this. Gives you an idea of this orthomosaic that we stitched together after that, and you can see peppered across this area a bunch of seals that are there for breeding purposes. In this case, we're sampling, like I said, about 2.5 centimeters on the ground resolution, which allows us to be able to identify pups from adults and things like that when we zoom in. I can give you, actually, I'm going to just jump out now and actually show you some of the data, if I can. And, go back out. So this is actually an island off of Nantucket. Oh, it's going to be in the wrong screen there. And so this island's about 3 kilometers wide and about 1 kilometer high. And if we zoom in here, pretty soon you'll start to see some seals appear. And as the high resolution imagery tiles in, you're actually able to detect individual pups and adults. And if we snapshot here, you can see some researchers actually working on a seal. We snapshot here. I'll let you know we flew this right around Valentine's Day, and you can actually see a couple of groups of seals mating right here. Right where we dropped off our equipment, good indication that we really didn't disturb the animals all that much. So that's the RGB imagery. I quickly showed you. Here's that same survey done with a thermal camera, and that really allows us to be able to detect seals, especially those pups that are white on the snow. They really pop. And if you look to the top left, there's actually an area of scrub and woods there that we can actually see seals that have come up inside the forest that we're able to actually count, improving our population assessments. This is a real pain to be able to go out and count seals, one by one. And so our next step was to actually move into a computer vision workflow where we take advantage of the temperatures of the seals and their sizes to be able to enumerate them rapidly. And so here's our workflow going from those thermal signals to individual seals. We bring in the thermal map, we select warm pixels, we convert those to polygons, and we do a polygon to pixel ratio to give us an idea of how many seals might be in that. We then parse that out and do some edge detection with a high-pass filter that allows us to see individual seals in the piles, and then we come out with some great imagery. So here's an example. This is what that looks like. We use a con tool to select those seals. We then build those convex hulls around the aggregations, measure those ratios for pixel temp, and actually look at the temperatures. It turns out the pups are a little bit cooler than the adults, which is also helpful for discrimination. And then we run that high-pass filter on the aggregations to be able to pull out individual seals. So this allows us to then move into population assessments that instead of taking 2 weeks in terms of counting and going to assessments, to doing it in 20 minutes. The other great thing about this is that individual analysts will always count different numbers of seals because of our perception differences. This technique does it the same way every time, which will give us more precision and better abundance estimates through time. If you look at our results, we see our detection model on the left there and our human-generated counts on the right. The model was tested on Hay Island, but then applied to Saddle Island, both with temperature correction and not. And you can see that we get very close to the human counts, giving us some confidence that this is actually a really great way forward for us. We're now applying this technique to things like penguins. So here, for example, it's a quick launch of an EB right down on the Antarctic Peninsula. I just got back last week, actually, from doing these surveys. This is going to be a fantastic way for us to be able to assess these penguin colonies. To give you guys an idea of what a penguin colony looks like at the Antarctic, here's a structure from motion video of Avian Island. It's about halfway down the Antarctic Peninsula. Where you see pink is the actual colonies of penguins and other seabirds. It's their guano, so they eat a lot of krill, so their poop is pink. And this gives you an idea of how barren that terrain is and a really good example of how those colonies are structured. Because the colonies are pink, it actually allows us to also do some multispectral work, which allows us to do very rapid assessments of animals. And so I'll give you an example of that. But when we apply the thermal camera, here's a thermal section of that island. The little inset at the top shows those individual hotspots that are individual birds. So we're able to take our seal counting technique and apply that to penguins in the Antarctic Peninsula. Multispectral map of that area, where you see that sort of middle gray with the black pepper inside of it, those are actually individual colonies of penguins, Adelie penguins on the island. Because that poop is a bright pink color, it really stands out in our false color near infrared images, allows us to be able to do multispectral analysis. When you talk to penguin biologists, they'll tell you that there's about 2 birds for every square meter in these areas. And this relationship is conserved across penguin colonies. This allows us to be able to do a very quick multispectral mission and to be able to estimate population abundance of penguins very rapidly just by doing the multispectral polygons. I'm going to switch now and take you to a coastal example here in North Carolina. This is a map that shows the Duke Marine Lab. The Duke Marine Lab is right up on this island right here. And we fly from the Duke Marine Lab pretty much every day. And we're very interested in this strip of sand that's essentially our last bit of protection from the opening inlet to the Atlantic Ocean. And for those of you who were around for the hurricane, this is what that looked like after the hurricane, as the waters from inland crested and flooded, sort of matching the sound side water level. You'll see that there's a bunch of waves that are propagating across the barrier island, essentially pushing material from the front to the back, which is pretty typical of what we would see. When we look at that orthomosaic and look at different structures, in this case a couple of trees, we lost about 2 meters of a scarp on that single event. And so very useful way to be able to look at rapid changes. Because we can do the 3-dimensional analysis, we'll also be able to look at elevation detection with our RTK drones and ground control points. We can actually do some really, really accurate measurements of elevation and change in this area. We were lucky enough to be able to do, before the hurricane, some comparisons with a terrestrial line scanner to look at our accuracy and precision in relationship to a traditional method. And so on the top there, we have an area that's vegetated, and then the bottom we have just a sandy area in comparison. And the first color map is a elevation map coming from the laser line scan. The one beside it is the UAV. And you can see that we've got some good agreement between the two. We have similar problems in structure from motion as the laser line scanner with occlusion from the plants. In this case, from structure from motion, actually tracking the movement of the grasses. But in the areas where we just have bare sand, we're less than a centimeter difference across these areas. So it gives us some strong confidence that we can use this technique to understand how these barrier islands are shifting in response to stresses like hurricanes. If we look at the data, top is before orthomosaic, bottom or middle panel is after after the hurricane, and the bottom panel is actually a change map. Where it's bright orange and red, we're looking at 2 meters difference, loss in elevation. Where it's green, we're looking at 2 meters increase in elevation. So this is a significant event on that island, and a really nice illustration of that. The one thing that's really interesting is you'll see in the back parts of the island where we have green, there's a lot of noise. And so we've been working on noise detection and removal algorithms to be able to deal with that. So when you look at the top panel there, that's an orthomosaic. And because that sand is so continuous, the structure from motion algorithm finds it very difficult to be able to find points to match. So, and that RGB mosaic shows that, how smooth and monochromatic the sand is. But what we can do is we can go in and using neighborhood statistics and standard deviations, we can actually find the artifacts and remove them and smooth them over and create a more accurate elevation model for comparison. When we look at a slice through that barrier island, we can see that the baseline was in blue. So before the hurricane, the hurricane whacked a lot off that island. You can see in orange. And now, actually, we've been surveying again, a month and a half later, we see significant recovery. So this island actually in many places is higher than it was before the hurricane event, which is really interesting. But we can see even with that, we're piling up sand on the backside of the island. Give you guys some context, that's why we're interested in this. This is one of our orthomosaics of Bulkhead Shoal right there on the edge of that strand. The background image is from 1993. And so we see this island transgressing towards the marine lab and be getting thinner and thinner. And so we're hoping that this tool is going to give us better resolution, higher resolution surveys, and better information for management. The last example that I want to show you is a Maritime Heritage one. And if you guys have ever been up the Potomac, right across from Quantico, there's a place called Mallows Bay. And Mallows Bay is a ship graveyard. After the First World War, about 200 Liberty ships were eventually ghosted there because no one wanted them. Those wrecks eventually burned to the waterline and remain there today. And this area is now about to become, I think, our nation's newest national marine sanctuary. What's really amazing is that those shipwrecks are turning into islands. So we actually have a really amazing intersection between maritime heritage and ecology. And they're now supporting actual ecosystems. I have another quick video that's going to zoom in. This is an orthomosaic from flights over that area. We're going to zoom in, and you'll actually be able to see some of these, especially close to the shore. They've actually become full islands supporting small mammal populations, insect populations, and vegetation assemblages. And then as we back off, and we'll slide a little bit over to the left, you'll see that these ships are stacked like cordwood in this bay, and are structuring not only the ecosystem above the water, but also below it. You can see submerged aquatic vegetation and siltation that's occurring. And so we're in the process now of vectorizing these and preserving them, so as we worry about things like sea level rise, we'll have access to this information in the future. So a really amazing sort of connection between ecology and maritime heritage made possible with the use of UAVs. So that's all I have. Running out of time, want a little bit for questions, but appreciate the time, and thank you.